Hi everyone. Today I'm going to share with you about the latest bioengineering enzymes using rational design strategy. So by definition, rational enzyme design is a strategy for engineering enzymes by predicting potential mutations based on the relationship between enzyme structure and function, then introducing those mutations through site-directed mutagenesis. So here's the question. Why do we need to engineer our enzymes? We know that enzymes have become very important tools in various fields, including pharmaceutical, chemical industry, and also the food industry. However, when enzymes are applied in industries to produce high value added chemicals, the substrates used are often not their natural substrate, resulting in very low enzyme activity. So we are going to discuss two approaches of bioengineering of enzymes to improve the stability and activity of enzymes for industrial application. One of the method is the multiple sequence alignment method, and another one is through the remodeling interaction network method. The first approach is using multiple sequence alignment. Enzymes with high sequence identity and structural similarity tend to have functional similarity. So based on this, multiple sequence alignment has been widely used in engineering enzymes to have improved properties. The very straightforward approach is to make mutations in the target enzyme to the corresponding conserved sequence of homologs with desired properties. One of the enzymes improved by using multiple sequence alignment method is amidase from Agrobacterium tumefaciens. Amidase is capable of cleaving the amide bonds present in the carcinogenic ethyl carbamate (EC) in alcoholic beverages. The amino acid sequence of amidase from Agrobacterium was aligned with three available urethanes, which is enzyme that also cleave the amide bond and shows ethyl carbamate EC degradation activity from Rhodococcus, Licinibacillus, and Candida. The catalytic triad of amidase, which is lysine at position 98, serine at position 173, and serine at position 197, was conserved in all of the sequences aligned. The conserved but different sites were identified from regions adjacent to catalytic triad and the conserved sites in the multiple sequence alignment. Finally, six mutants were designed based on sequence alignment, and the base mutant, which is the glycine convert to alanine at position 195, improved the activity of amidase by 4.9-fold. The improvement of this amidase enzyme could help the brewery industry to reduce EC levels in alcoholic drinks, making them safer for consumption. The second approach is remodeling interaction network. The active site of enzyme is the core region where the complete catalysis cycle is performed, including substrate binding, formation of the transition state, and then product release. The interactions such as hydrogen bonds, hydrophobic interaction, and salt bridges formed between the substrate and the enzyme active site play a crucial role in anchoring substrates to a correct position ready for catalytic reaction. It could be a useful strategy to rationally remodel the interaction network involved between the substrate and enzyme active site to modify the substrate binding affinity and hence enhance the enzyme activity. One of the enzymes engineered using this remodeling interaction network method is glutamate dehydrogenase from the bacteria Clostridium difficile. So this glutamate dehydrogenase enzyme was engineered to have improved activity toward non-native substrate, which is 2-oxopentanoic acid (PTO). But before that, let's just observe what will happen when the enzyme bind with its natural substrate, which is alpha-ketoglutarate. So when glutamate dehydrogenase bind with alpha-ketoglutarate, two salt bridges are formed between the gamma carboxyl of alpha-ketoglutarate and two residual of the enzyme, which is the lysine at position 71 and arginine at position 187. So they are relatively stable, and that is not our problem. So if you notice, the relatively stable structure formed by the enzyme glutamate dehydrogenase and alpha ketoglutarate through the salt bridges is because that the arginine at position 187 and lysine at position 71 are both polar. But if you look at our desired substrate, which is non-native 2-oxopentanoic acid (PTO), it actually lacks the polar group at the gamma position. Therefore, it would not form any electrostatic interactions with the polar residue, which is arginine and lysine. To resolve this problem, the residual lysine and arginine were hence mutated to hydrophobic alanine to modify the polarity of the binding site. This is done by the site-directed mutagenesis. As a result, the variant lysine converted to alanine at position 71 can improve the enzyme activity by 2.3-fold compared to the wild type. And this modification is significant to produce L-novaline, a non-natural amino acid with biotechnological and pharmaceutical applications. In conclusion, bioengineering technology using rational enzyme design helps improve enzyme performance by targeting specific structural features. Approaches such as multiple sequence alignment and remodeling interaction network make enzyme more efficient as seen with amidase with safer alcoholic drinks and glutamate dehydrogenase for producing useful compounds like L-novaline. So that's all for this video. Thank you for watching.